Good afternoon, everybody. Honorary Chairman, dear members of the organizing committee, ladies and gentlemen, it's my great honor to attend the third International Conference on Molecular Modeling and Spectroscopy taking place at the National Research Center in Cairo, Egypt. To our big regret, the pandemic around the world is still severe, and we are forced to hold conferences in a distant format. But I am confident that the pandemic is unable to stop the progress of science and organization of scientific international and national conferences. So thanks once more to the organizing committee for their efforts to organize this conference. And let me now start my talk devoted to the complementary Raman and Neutron scattering studies applied to biomedical tasks, the title of which you see on the screen. In more details, it comprises two items. The first one deals with the investigation of competitive effects of cholesterol and melatonin embedded in model lipid membranes. And the second one deals with the search of Raman markers of NETosis, a special form of programmed cell death. As for a brief introduction, I would like to say that we know very well that the cell membrane is a complex and dynamic biological structure composed of various lipids, sterols, and proteins, whose primary functions are setting a boundary between the cell interior and external environment with selective permeability. Addition of various molecules to the membrane can change its physical properties and alter its structure, which in turn may affect its functionality. Thus, it is crucial, to our opinion, to know the origin of these changes and understand the intramolecular interactions responsible for embedding various additives to the membrane, e.g. cholesterol and melatonin. Our choice of melatonin and cholesterol as an additives was not accidental. The cholesterol, for example, is traditionally linked to various dysfunctions as a trigger, while melatonin's protective role has been claimed in cardiovascular, Alzheimer's, and age-related diseases. The role of lipid membrane changes in Alzheimer's disease has been proposed as a trigger of amyloid toxicity. Thus, the role of membrane-active molecules such as cholesterol and melatonin and how they alter lipid membrane properties is of a great importance in understanding molecular mechanisms of membrane-mediated interactions and associated diseases. Thus, we consider that it is appropriate to focus on three-component systems to search a relationship between cholesterol, melatonin, and phospholipid, and reveal their common influence on the structural and dynamic properties of membranes using Raman spectroscopy, small-angle neutron scattering, and small-angle neutron diffraction. But let me also mention that cholesterol and melatonin incorporate into the lipid bilayer at different positions. It has been proposed that melatonin resides in the head group region of bilayer and thus makes it more fluidic. On the other hand, cholesterol incorporates parallel into hydrocarbon chain region and its hydrophobic head close to the lipids head group. Such localization of cholesterol causes the membrane to become stiffer and thicker in the transverse direction because of the increasing order of hydrocarbon chains. Now I am coming to our experimental results. Sans measurements were performed at UMO. Some of you are familiar with this facility at Dubna at IBER-2 pulse nuclear reactor. And the obtained scattering curves for DPPC are plotted in the top left figure. The accessible Q range makes it possible to evaluate an overall size of unilamellar vesicles in the low Q region, and length scales relevant to the membrane itself in the intermediate Q region. The smoothness of the latter part lends the data well to evaluating the membrane thickness via fitting approach analysis. As for the sound measurements on the right side, you can see a figure with a typical diffraction pattern for DOPC lipid with 9% cholesterol and 9% melatonin. The phi here is the detector angle, which is always positioned at 2 theta, where theta is the sample angle. These curves reveal typically up to 6 diffraction peaks that were fitted to the Gaussians. The obtained data was processed in a certain way via Fourier transformation, but let me skip the details of that, as it is a long story. So this processing finally results in neutron scattering length density profiles, NSLD, representing the membrane special structure in a transfer direction that can be used to determine the thickness parameters. They are shown in the two bottom figures on the left and on the right. Let me comment these results in a bit more details. If you look closely to the NSLD profiles, then intriguingly, the NSLD profiles suggest a difference between the interactions of cholesterol and melatonin with demonosaturated DOPC and fully saturated DPPC lipids. Tables at the right side compiled experimentally determined bilayer thickness parameters. And you can see that the thickness of the three-component DOPC-based bilayers increases only with the addition of 29 mole cholesterol and 9 mole percent melatonin, while it decreases in all other cases relative to the NETosis bilayer. So you can see here marked in the blue the only position where we have increase in comparison with the NETosis bilayer. But in the case of the DPPC based bilayer, the upper table, on the other hand, thickness bilayer thickens with the addition of cholesterol and melatonin in all cases of ternary system studied in here. Yes, you can see that in all these positions we have increase in comparison with the NET-DPPC bilayer. Nevertheless, I'd like to mention that the effect of cholesterol is more or less similar to some extent on the saturated and unsaturated lipid bilayers, and it is in perfect agreement with recent conclusions and studies of the other authors. At the same time, the similarities between DPPC and DOPC-based system end with the addition of the third component, melatonin. The figures here demonstrate the relative thickness changes of bilayers prepared of A, DPPC and B, DOPC as functions of the molar fractions of cholesterol on this slide, and on the next slide you will see the same figures for melatonin. So some important results are shown, are indicated on the slide. Yes, the increasing the cholesterol concentration to 29 mole is suggested to increase a mean value of bilayer thickness up to more than 3 angstrom for pure DPPC and pure DOPC membranes. Addition of melatonin to DOPC membrane leads to decrease the cholesterol condensing effect. And here you can see the fluidizing effect of melatonin. Interestingly, the effect of melatonin, when analyzed in a similar manner as that of cholesterol above, is as much as reverted, depending whether the additional cholesterol is present in DPPC bilayer or not. While the increasing concentration of melatonin causes the thickness of DPPC bilayer with no cholesterol to decrease, it remains unchanged or slightly increasing in the system loaded with cholesterol. This again reaffirms the prevailing interactions between the DPPC and cholesterol that quickly overcome the interaction between the DPPC and melatonin. However, melatonin is known to ensure a bilayer thinning due to its partitioning amongst the lipid head groups, increasing thus the bilayer surface area when added in the case of two-component system. So this is for the neutron scattering results, and now I'm going to the second part of the item number 1, the complementary results on Raman spectroscopy, which is known to be a sensitive instrument for probing conformational changes manifested in the Raman spectra of biological samples under certain conditions. Obviously, we started from the measurement of pure DPPC Raman spectrum and probed signals in the 1600 centimeter inverse range of vibrational frequencies. And it's well known that the spectral range between 1030 to 1150 is sensitive to trans-gauge conformers, which can serve as a spectroscopic markers indicating the phospholipids, hydrocarbon chains, ordering and disordering. The Raman modes 1127 and 1062 in the spectrum characterize asymmetric and symmetric vibrations of the carbon atoms and are known to originate from the trans structure. This was studied earlier. The Raman line at 1096 centimeter is associated with the sensitivity with a single gauge defect and with the formation of various gauge isomers. Hence, the ratio of the intensities of the spectral weights at 1096 and 1127 and 1096 and 1062 can be used to indicate the ratio of gauge trans isomers of DPPC molecules. This slide demonstrates the preferential areas of influence of melatonin and cholesterol. As I already mentioned, that their localization in the lipid bilayer structure goes in a different way. You can see the area of impact of melatonin and influence on the two Raman bands, and the main influence of impact of cholesterol on the left trans band. Those are the results for two-component system. You see the normalized Raman spectrum in this range from 1040 to 1140. As I mentioned, sensitive to the trans-gauge conformations. And the input from the carbon-carbon stretching mode of the primary metal groups of the hydrocarbon change is best characterized by the ratio of the spectral weights, as I mentioned, 1127 and 1096, while the increasing occurrence of gauge conformers is best evidenced in the ratio of spectral weights at 1062 to 1096. From these graphs, this one is DPPC plus cholesterol, and this one is DPPC and melatonin. It's clearly seen from these graphs that with increasing concentration of cholesterol on the left figure, we see an increase in the intensity spectral weight of transconformers here at this Raman band. A similar picture we observe for melatonin, right figure. Although the rise of the Gauge conformer is here, the Gauge conformer is more moderate in comparison with the cholesterol. This is in a good agreement with the neutron scattering results. Now I would like to demonstrate you the quantitative characteristics of the ratio of the spectral weights as a function of the content of cholesterol and melatonin in DPPC bilayers. The first row, for example, let me briefly comment this table. The first row in the upper table here shows an approximately twofold decrease from 1.75 to 0.92 in the ratio of spectral weights 1062 to 1096 bands upon loading the neat DPPC bilayer with the melatonin at concentrations up from 9 to 29 molar percent. This indicates a membrane fluidization, let's say, in its hydrocarbon chain region. A more moderate decrease in the ratio of spectral weights at 1127 to 1096, the first row of the bottom table. You can see that it's more moderate than here. This decrease is associated with the incorporation of melatonin mainly into the head group region of the lipid bilayer, weakly affecting its hydrocarbon chains. The situation is just opposite in the case of the cholesterol effect, which is best detected by evaluating the ratio of spectral weights, as I mentioned, at 1127 to 1096. We can compare the first columns of the upper and bottom tables here and here. You can see here we have rise for 2 folds, and here it's much more moderate. And at the final part of item 1, I would like to demonstrate you really the complementary between the Raman studies of this task and neutron scattering for the 3-component system. This one is for the case with added cholesterol. Here, Raman results for 3 different molar percent of melatonin. And this one is the complementary results of Raman and Sons for the other 2 Raman bands, 1062, 1096, and here it was for 1127 to 1096. I'm concluding this part of my presentation by this summary to this item number 1. Let me quickly read it. The impact of cholesterol and melatonin is opposite and competitive in the case of 3-component system of lipid, cholesterol, and melatonin. And dear colleagues, I would like to mention once more that this was the main purpose, main aim of our studies, because in the case of 2 components, for example, lipid and cholesterol or lipid and melatonin only, was studied very broad by many authors, but three-component system was studied very poor. So specifically, cholesterol induced an increase in bilayer thickness, while melatonin induced a decrease in the bilayer thickness. Raman spectroscopy demonstrates high sensitivity to the alteration of the trans-gauge conformers changes in the three-component system. And thanks to the developers of our Raman spectrometer, which is very sensitive to this sort of experiments. The dynamic properties of the studied system indicate the predominant influence of cholesterol ordering effect rather than melatonin, and we observe this both in Neutron measurements and in Raman also. Although at the high concentrations of melatonin, about 20% and higher, the fluidizing effect of melatonin is capable to inhibit the condensing effect of cholesterol. The demonstrated condensing effect of cholesterol and the fluidizing effect of melatonin appear in an additive manner upon their mutual presence. So let me now pass to the second item, which to our opinion is very attractive for study. And it deals with the signaling pathways and possible Raman marker of NETosis. As I mentioned, it is a special form of programmed cell deaths. And there are two main forms of NETosis, classical or suicidal, and the second one a vital one. But today we will consider and speak a little bit about the classical NETosis. Firstly, I suppose there is a sense to give some brief general information on neutrophils, which are the largest population of leukocytes, which normally comprise up to 70% of all white blood cells. Neutrophils differentiate in bone marrow from hematopoietic stem cells and after full maturation enter the bloodstream. And they circulate in bloodstream for no more than 72 hours. But the very important moment is the third item. So when the host organism is infected, neutrophils immediately migrate from the bloodstream to the site of infection, providing a first line of defense against bacterial, fungal, and viral infections and pathogens. This is very important, and it's comparably new statement that the neutrophils are the first line of defense against pathogens. And all this began in 2004. So since the decoding of the genetic code in the mid of 1960s and until 2004, it was considered that DNA serves exclusively for the storage of genetic information. We were thinking so. However, it was found that the chromatin is also part of innate immunity and performs important effector functions against a large variety of pathogens. And this discovery belongs to a group of scientists working under the guidance of Arturo Zielinski or Zylinski, and who published their, together with Brickman, revolutionary work in Science Journal in 2004. They have considered that neutrophils were able to kill pathogens outside the cells by releasing chromatin fibers or so-called neutrophil extracellular traps, NETs. Since neutrophils lost their viability in the process of trap formations, Steinberg and Greenstein denoted this form of neutrophil cell death as NETosis in 2007. So NETs are a modified chromatin decorated by bactericidal proteins of granules, nucleus, and cytoplasm. Various pathogens such as bacteria, fungi, viruses, cytokines, and other physiological stimuli can cause NETosis. Here you can see two nice videos. The left one for phagocytosis by neutrophil cell, and on the right side, formation of NETs after activation. The mechanism of classical NETosis is shown on the figure, and provided this picture is provided with the kind permission of the author, Dr. Nina Vorobyova, from Moscow State University. And on the right side of the slide, you can see a list of the main components and organelles involved in NETosis, among which one of the most important is NAT oxidase, or NOx, which is a membrane-bound enzyme complex and is one of the major sources of cellular reactive oxygen species. But I would like to say that the schematic, this is just the schematic presentation, but the mechanism and signaling pathways is a long story, and probably next year at the third international conference, I will give for your attention a special talk on NETosis in more details. But now I would like to mention that along with the NAT oxidase, another very important component taking part in NETosis is peptidyl arginine deaminase, so-called PAD4, which provides citrullination of histones, because PAD4 also transferred from the cytoplasm into the nucleus, and this leads to a weakening of the connection of histones with chromatin, and its subsequent decondensation. So it's entered the nucleus, and this results in chromatin decondensation. After the destruction of nuclear and granular membranes, electrostatic binding of granule contents to the condensed chromatin occurs. And finally, at the final stage of the process, chromatin is released into the extracellular space, and NETosis occurs. So in our first set of Raman experiments on NETosis, we focused our attention whether we can detect a Raman band of citrulline in the spectrum, Raman spectrum, and I'm happy that we succeeded. So you can see here the main our results on Raman spectroscopy. We revealed the evolution groove of the citrulline peak. Here you can see it. The red one is the control. The blue one is after 30 minutes of activation, and the yellow one after the 40 minutes of activation. So this allows us to say that the Raman spectroscopy really can serve as an early diagnostic tool, indicating that the citrullination of histones started, and this is the pre-NETosis situation. This was published in 2020. And now at the very final stage of my presentation, I would like to say that now we are on the way to study NETosis with sterile activation using UV and blue light. This will be done by our Raman spectrometer and immunofluorescence microscopy. And on this slide, you see our present participants. So it's Joint Institute, Moscow Regional Research Clinical Institute, Moscow, our partners. It's Institute of General Physics in Moscow, University of Belgrade, Serbia, Shafarik University in Košice, Slovak Republic. And I'm taking this opportunity to invite, to call our Egyptian partners from research, medical organization to join us, to cooperate with us in this task as well. So that's the end of my presentation. Thank you so much for your kind attention. May everybody be healthy. And I would like to thank once more the efforts of the organizing committee of this conference. Thank you very much. Bye.